and she was at Brown University for a number of years before joining the faculty at Davis. And she's recently retired, but continues to do research with Emerita status. She is really a leader in the field of plant ecological genetics and genomics and evolutionary ecology. She's had numerous awards and publications. She's a past president of the American Society of Naturalists and the Society for the Study of Evolution, a member of the National Academy, and has done a lot of interesting work on a lot of different species. But today she's going to talk to us about how natural variation in the seasonal germination niche shapes plant life histories and climate adaptation. Thanks, Andy.
Well, thanks, Chris. It's been really a fun virtual visit so far. I've enjoyed talking with everybody, and I appreciate the opportunity to be with you by Zoom. So I'm going to start by acknowledging the sort of major co-authors on the work that I'll be presenting today. On the Arabidopsis side, I'll be talking about work spearheaded by my former postdoc, Alejandro Martinez Verdeja, and Michelle, Miki, and Liana also contributed to that work. And then I'll be talking about work, a collaborative work with and a group of plants that you're going to hear more about later, the Streptanthus clade. And so acknowledging all of my co-authors. You'll meet them more later. So all of this work is motivated really by what I think is a fundamental question in the 21st century, which is whether or not plant populations will be able to adapt fast enough to keep up with rapid climate change. So if we assume that plant populations adapted in the past to be well matched to their climate, perhaps along an elevation, climate gradient like we see here, then when climate change sort of pulls the rug out from under them, it means that populations are no longer adapted to what they're experiencing. And with rapid climate change, these lags could be extreme. So Sally Aitken worked out for Sitka spruce that predicted warming would lead to, for the adaptive trait of bud set, an adaptational lag of like 50 days. So will populations be able to persist under these conditions? And a related question is whether or not phenotypic plasticity can help. So for example, trees are bursting bud earlier in present day conditions some 100 years ago to warming springs. Will that help populations to persist? So I'd argue that seasonal timing of life history events is particularly critical for climate adaptation. So for example, this Arabidopsis flowered at the wrong time, and that was probably a very bad decision for it. And so to match life history events with favorable climate conditions depends on a variety of plastic developmental responses to predictive seasonal environmental cues. So these might include ambient temperature or the accumulation of degree days or heat sums, soil moisture, resource availability. Day length is an important cue, or winter chilling we'll talk a lot about today. But a key point is that climate change is now altering seasonal environments. It's not just overall climate warming, but what does it mean for the different seasons? So these are average monthly temperatures 100 years ago, and getting warmer as we move up to this is when this was downloaded. And so what does that mean? It means winters are getting warmer, which could reduce chilling cues for vernalization. Springs are coming earlier, autumns are coming later, so the growing season is altering. Summers are getting hotter, so heat and drought stress are increased. And in addition, there are changes in the amount and timing of precipitation. For example, in California, as I'll talk about more later, the onset of the rainy season is now nearly a month later currently than it was from 1960 to 1989. there's also greater climate variability. And so what this means is that there may be a mismatch between the developmental cues that plants use and the new seasonal environments. And so a big question then is, will plasticity to these cues be adaptive or maladaptive? And can these cue responses evolve? So I'm going to focus on germination timing, which is critical for fitness and adaptation. The timing of germination determines the environment a seedling will experience. October is warm and long days compared with December, for example. It also determines the biotic cues, and it also determines environmental cues for later development, like photoperiod. And so, as you'll see, it can affect life history, phenotypes, and fitness through growth, timing of reproduction, and survival and fecundity. And importantly, the optimal timing for germination may depend upon climate. So, for example, in great work out of Björn Agrun's lab, in Italy, there's selection for later germinated or more dormant type in the field in Italy, whereas in Sweden, selection favors less dormant, early germinating genotypes under Swedish conditions. Okay, so when I talk about the germination niche, I mean the combination of environmental conditions under which seeds can germinate. And to do so, seeds use these environmental cues to release dormancy and germinate under favorable conditions, or to avoid, or dormancy to avoid unfavorable conditions. So again, light could be important, ambient temperature, soil moisture. There needs to be enough water for the seed to be able to germinate. But in addition, the accumulation of hydrothermal time, and particularly what has been well studied is the release of dormancy by summer heat, what we call after ripening. And another important cue is the release or induction of dormancy by winter chilling, what we call stratifications. And so again, in different climates, you would expect different cue responses to be favored to promote appropriate germination under favorable conditions. So today I want to talk about two big questions. First of all, just looking at natural variation and climate adaptation and seed responses to chilling cues. And secondly, thinking about the germination niche and its response to the changing timing of the rainy season in Mediterranean climates. And so I'll be talking about two study systems today. First of all, Arabidopsis thaliana, which my lab's been working on many years, and a newer collaboration I've mentioned on the clade of Streptanthus and Colanthus and the Brassicaceae. These are native California wildflowers, and really marvelous clade. Well, let's start with Arabidopsis, and just, for a plant biology department, I don't need to really review the virtues, but there are many, many tools like mutants and near isogenic lines for studying candidates. Importantly for this work, there are stock centers where you can have worldwide collections from diverse climates, and it has a worldwide pan-Eurasian distribution. There's mapping lines we can take advantage of, and importantly, this was the first species to have its genome sequenced, plant species. There have been a lot of resequencing, and so that enables us to do genome-wide association studies, but also to look at associations with environment and the climate of origin and particular genotypes. Now, the mechanisms of flowering and germination response to environmental signals are very well studied in this species. And people have identified, using QTL mapping and genome-wide association, major functional variants that underlie natural variation in life history timing. I'm going to focus particularly on seed dormancy and germination timing, and you'll hear about this gene DOG1 later on in the talk. But Arabidopsis thaliana has also emerged as a really interesting ecological model, or evolutionary ecology model. what's useful about it is that because it's an inbreeding annual, it's possible to replicate genotypes across different experiments and then reference them back to things like the climate and the site of origin. There is a clear signature of a genomic signature of climate adaptation in these worldwide collections. And there's been a lot of mapping, QTL mapping, GWAS in field experiments by us and some other groups. And what's great is that it really does have a wide climate distribution from Mediterranean to really, Nordic, subarctic conditions. And across this range, it exhibits a really wide life history variation. So, for example, this is kind of a heritage slide from my lab that I can't bear to let go of after. But basically, this illustrates different the timing, seasonal timing of events in Thaliana across different parts of its range. So in Mediterranean regions, it germinates in late fall with the onset of rains, has a short growing season as a rosette, and then as spring comes, it transitions to reproduction and sets seed before the onset of lethal summer conditions, which it survives as a dormant seed to germinate the following spring. So that's a winter annual strategy, but a fairly short one. And in contrast, in Scandinavia, you'll see germination in late summer and fall, surviving under the snow as a rosette for a really long period of time, and then flowering and setting seed the following spring and summer. So the growing season is completely flipped from across the two ends of the range. Now you can also have spring annuals that overwinter as seeds and germinate in the spring. You can have rapid cycling genotypes that germinate several times a year, and these are common in Central Europe in particular. And you can also have winter annuals that, again, germinate in fall and flower in spring. So a wide range of life history strategies. And so it's really the induction and release of seasonal dormancy that shapes this life history variation. So in a typical winter annual, here are the seeds being dispersed in spring, and then they would overwinter over summer, and they're exposed to heat, and they after ripening, and that releases dormancy, and so they then might germinate in the fall and then go through their life cycle and release seeds the following spring. So that's a typical winter annual life cycle. But spring annuals might remain in the seed bank through the fall and the winter, and they often require a long cold stratification in order to break dormancy, and so then they would flower the following spring. You might see these in montane habitats, for example. And then rapid cycling might have very low dormancy, low dormancy, so they could germinate in fall, they could germinate in spring. You can have multiple generations per year, as you can see in this English agricultural population. So there's a lot of work showing natural variation in primary dormancy and after ripening, and that shows clinal variation with climate, with higher dormancy in warmer southern latitudes. But there's less known about variation in response to winter chilling, and that's a little bit more complicated. So first of all, brief cold stratification, like here, for example, could break primary dormancy and induce autumn germination. But on the other hand, spring annuals require a longer stratification in order to germinate, and they don't germinate till after winter. But in some genotypes or species, in fact, prolonged cold exposure induce secondary dormancy, which prevents germination during winter and spring. So all of those responses to chilling are possible. So here, I want to, first of all, ask, what's the genetic architecture of natural variation and sea chilling responses? Does variation in this aspect of the germination niche co-vary with other life history traits to shape life history syndromes? And then finally, do genetic variants or phenotypes underlying this germination niche trait exhibit a geographic signature of adaptation to climate. So this was a huge project spearheaded by Alejandra, where she mapped these responses to seasonal chilling cues. She did this using a wide panel of Arabidopsis accessions that had been sequenced by the 1001 Genomes Project. And so she calculated a bunch of phenotypes, did a genome-wide association, and then Michelle Stitzer then did a haplotype analysis of a candidate gene, which gave us some interesting perspectives. And then we did geographic and climatic associations. So the parents for these seeds were grown under spring conditions, simulating a winter annual situation. And Alejandra measured days to flowering and days to senescence on these parents. And then as each plant senesced and produced fresh seeds, she immediately put them into her pipeline and did germination assays with these fresh seeds in the different chilling environments. Also did a separate assay for after ripening requirements that I won't really talk about here. A massive experiment. So there were then 13 different stratification treatments, a range of chilling lengths and 2 temperatures. And then those were all placed into inductive conditions and scored for germination after 7 days. This consumed the lab for 1.5 years. And there was a team of around 10 undergraduates helping to collect the data. It was a lot of fun. And what all this came down to was that she identified 2 major axes of variation in germination responses. So from all those different treatments, and I'll break this down for you in a minute, but there's one axis explaining a lot of the variation that's just genotypes that had low germination, on the other hand, genotypes that had high germination. But the second axis was very interesting because it distinguishes genotypes where a brief cold exposure releases dormancy, the genotypes where long cold induces secondary dormancy, so kind of opposite reactions. Notice, by the way, there are purple and green on this slide, and you will see in a few slides what that's about. Okay, so just breaking this down a little bit. So if we look first of all at the low end of PC1, we can see there's a class of genotypes that are don't germinate very much in all conditions. On the other hand, there's some that are just pretty promiscuous and germinate all the time, regardless of chilling treatment. And then in the middle, we have genotypes that, on the one hand, germinate pretty well. And then as they're exposed to longer durations of cold at 4 degrees, they go back into secondary dormancy. And on the other hand, the purple end, they actually are induced to germinate and remain able to germinate even after exposure to cold. So kind of the opposite reaction. So that's kind of interesting to see that. So we can think of this ecologically. The seeds with overall levels of germination are likely to remain dormant in the seed bank. This would be what we call a bed hedging strategy to ensure at least some germinate in good environments. And then these are likely to germinate anytime, all the time, as long as they experience appropriate moisture and light conditions. And then secondary induction by cold of dormancy results in a winter annual life history. They can't germinate in spring. Whereas this strategy allows germination in fall, but also in spring. So it widens the seasonal germination niche, this possible. So genome-wide association analysis associated SNPs associated with dormancy and chilling response. There are several candidate genes associated, but the big one was our old friend DOG1, which was already well known as a major functional variant for the candidate gene, was actually tagged by the highest scoring SNPs for PC2. So this is all had been previously known as validated. It's a 3-base indel, and the D haplotypes lack self-binding ability. So it's definitely a major functional mutation. So Michelle Stitzer basically took published sequences to reassemble the dog1 exon 1 to look at these haplotypes. And so she found, first of all, there's an ancient divergence of these haplotypes. And you'll see more about that in a minute. She also identified 2 new haplotypes falling within this green clade, ECCY and then ECSY and EFSY. So, and we'll talk about those more in a minute. But if you look at these top SNPs for chilling response, what you see is they're differentiating blue and purple genotypes. In other words, they're clearly differentiating the different haplotypes. And in particular, the D show less secondary dormancy induction, and the E haplotypes have a more narrow niche winter annual life history. These guys could also germinate in spring and have a broader seasonal germination niche. Okay. So if we look at the SNPs associated with chilling responses, we can compare those to the sort of distribution of randomly chosen SNPs. And what we find is there's a signature of climate adaptation, especially for PC2. So this is the mean, the turnover with the temperature of wettest quarter. This is using gradient forest analysis, looking at allele frequency turnover along climate gradients. And this is compared with just randomly chosen selected SNPs, which show a lot less climate turnover. So there's basically a strong association of the SNP alleles associated with these chilling responses and temperature of the wettest quarter, which in part distinguishes Mediterranean climates. And in fact, if we look at the dog haplotypes, we see they have different climate distributions. And in particular, the purple haplotypes, the Ds, inhabit wetter summers. And some of the D haplotypes are limited to Mediterranean climates with very dry summers. And then if we look at these ancient haplotypes, we see very strong geographic patterns. So ECCY is the ancestral haplotype, and that's widely distributed in Eurasia. And it's the only haplotype found in Africa. And so there are some ECCYs here in China that are a little harder to see. They're here. And then the DSY is the light purple, and that's widely distributed in Eurasia. And then the dark purple DRY is distributed in Iberia and kind of Western Europe, and also is kind of the dominant one in North American accessions. So this might be a founder effect, or it might represent where they were introduced from in Western Europe, or it might have something to do with their invasive species lifestyle. So it's interesting to think about all of this in a sort of paleo-ecological perspective, and realize that the climate niche for Arabidopsis thaliana has moved across the landscape with glacial cycles. So Zhu et al. Kind of hindcasted the predicted climate space for thaliana during the last interglacial, and you can see it was distributed across across Africa to China and Europe here. And then they contracted into glacial refugia, and then came out of the glacial refugia during the Holocene 14,000 years ago or so. So here we see them coming out in Europe, coming out of the glacial refugia and kind of mixing across Europe as they move northward. And so what Michelle's analysis revealed is that the origin of the dog1 haplotypes and their diversification occurred during periods of climate change. So the ancestral ECCY allele, then the split of the E's and the D's occurred a long time ago during a glacial period, 365,000 years ago. But then the SY and the extant D lineage is diversified later. DSY during the last interglacial, and the DRY actually during the Holocene warming, although they'd split much earlier. So it suggests that these things split, and then there's a lot of contraction and extinction, and then when good conditions come again during the interglacials, there's a rapid diversification and spread. Okay, and then the two endemic Iberian ones also arose during the last glacial period and remain in Iberia today. So what's interesting is that the D haplotypes, which are widespread, the light purple ones across Eurasia, it's thought that about 40,000 years ago, there was a kind of and possibly more recently, there was this spread from Central Asia, sort of westward and eastward, what are called the non-relict populations of Arabidopsis. They kind of dominated, especially agricultural lands in Europe. Well, the D haplotypes, the rel are much more common in these non-relict genotypes. And so what we're speculating is that possibly the broadening of the germination niche to include spring germination might have helped contribute to post-glacial expansion, especially into agricultural environments. So one other thing, when we look at the adult traits, we see that there's a strong correlation of chilling responses with flowering time and senescence time. So late flowering and senescence are correlated with secondary dormancy induction by prolonged cold. That would be the winter annual strategy here. And on the other hand, this is kind of a broader seasonal niche of early flowering and could germinate in spring or fall. So this might be more of a spring annual or rapid cycling strategy. So we wanted to know whether this could be pleiotropic. And in fact, dog1 is associated with flowering in other genotype in GWAS studies, but not in our experiment. And so, in fact, what we found was completely different loci were driving the flowering time phenotypes that we observed. And so that suggests that maybe these syndromes are not just pleiotropy at one locus, but in fact, the result of correlated selection on different loci across the genome, which is interesting to think about. So when Moy Exposito Alonso wrote a commentary on this paper for PNAS, he actually took Alejandra's data and mapped them on the landscape and characterized them into sort of a spectrum of 4 different life histories, from the Mediterranean rapid cycling, high dormancy, rapid life cycle, to the Scandinavian winter annual, late flowering only, and then the rapid cycling in agricultural regions in between. So it's kind of, you can kind of think of this, these syndromes kind of dispersed on the landscape and correlating with climate. Okay, so a little bit about streptanthus in this part, which is looking at one species of streptanthus, tortuosus, which can range from being annual to biennial or perennial. And here we're looking at low elevation that's a low elevation population. Here's a high elevation population. At low elevation, they germinate with the first fall rains. At high elevation, after snow melt in the spring. So here, this is days of the water year, and the low elevation population immediately germinates in fall, and then the high elevation population is the following spring. So we wanted to know whether this ecological variation could be mediated by population variation and germination queuing. And so we did an experiment with different levels of stratification, simulating winter chilling. And what we found was a strong elevational decline in seed chilling requirements. So this is the elevation of source populations. I should have said that these were collected from populations across an elevation gradient across the Sierra. And then as we have greater and greater amounts of chilling after 7 weeks, the decline completely disappears and all of the seeds will germinate, or many of the seeds will germinate. So that's suggestive of adaptation to climate through evolution of seed chilling requirements and Streptanthus too. Okay, so now I want to switch gears and talk about the germination niche in Mediterranean climates, and particularly as the Mediterranean climates are changing across the world. So a key feature of Mediterranean climates is the very wet winters, as in Oroville, California here, and hot, dry summers. And so for plants in these environments, the rainfall comes starting in fall, and at least for winter annual plants, they germinate in fall with the first fall rains. And then they grow during the winter, flower in the spring, and then summers are very dry and hot, and so there's a lot of drought stress. And so for annual plants, the solution is to overwinter or over summer as dormant seeds. And for perennial plants, then they have to dealing with stressful dry conditions. There's a lot of interannual variability in rainfall, and this is fall rainfall, so in the timing of rainfall also. And in addition, there's an ongoing trend that was recently identified. So this is autumn rainfall. There's less falling in the autumn, and it's coming later. And this is in California. So this could have potential consequences for winter annual plants in California. So we looked at this in Arabidopsis thaliana, and this was a genetic experiment that turned out to be more of an ecological experiment in the end. But this was a huge screenhouse experiment. It was spearheaded by Alejandra Martinez Verdeja again, where we manipulated the onset and duration of the rainy season under ambient seasonal temperatures and light. And there were 2 experiments, actually. One was dispersing the seeds at monthly intervals that would simulate different rainfall onset dates and scoring the timing of each individual seed. And then the second was to manipulate the timing of germination, force seeds to germinate at a particular time, and then measure the phenology and the fitness of different cohorts. So here, for example, and we also vary. So for example, in the September cohort, then we would have different lengths of season, in other words, different onsets of the summer drought. And so there were a lot of treatments here, including a drought treatment I won't talk about today. And we did this with 4 multi-locus near-isogenic lines, or NILs. And so these were factorial combinations of natural functional variants of DOG1. You've already heard about that. And also the flowering time gene, Frigida. And I won't talk much about Frigida today, although it did have some effects on flowering. So for DOG1, what we found is that it does have a significant effect on germination, but only in certain cohorts. So we're just zeroing in right here. So freshly dispersed seeds in spring in April through June, you can see that the green and the purple bars are farther to the right. So this is germination date. This is actual germination timing of each individual seed. And this is the cohort in which it fall for the first time. And the main thing to notice here really is that for these DOG1 genotypes, some of the seeds delay germination until the following fall, which is the normal rainy season. So dog one only has effects during this narrow seasonal window under our conditions on germination timing, but it does have ecological consequences. So then in the second experiment, we could measure fitness. And what we found was, and here, these are the different onset of drought here. These are the seasonal cohorts, and this is fruit number. And for all of the genotypes, there was a pretty similar pattern that fitness was highest in October and then declined rapidly as rainfall came later. And also in spring and summer, germination was pretty lethal, but especially for those with, well, you're going to see this in the next slide, actually. So we'll see the effects of the different genotypes. So then what Liana, our collaborator, did was to combine the data from the two experiments. So taking for each individual seed, saying this is the date it germinated, then we have these fitness functions from the second experiment. What would it be? What would its fitness be? Projecting the fitness. And then using that to calculate for all of these different rain onset cohorts, what fruit number average would have been under different drought onset treatments. And so we see a couple of things, as we saw before, the later rainfall onset and the early spring drought both seem to reduce fitness. So, and this is the historic mean rainfall onset for California. This treatment is the current conditions. And you can see that that would result in a significant fitness decrease for all of the genotypes. And, you know, lower fitness in March than in April for most genotypes. Where do we see fitness differences? Well, in particular, you see fitness differences for spring rains, where there's sort of zero fitness for the ones without dormancy, and the dormant dog one genotypes do manage to eke out some fitness because some of the seeds stay dormant and wait until the fall rains before they germinate. Okay. So basically the idea is you can combine data on phenology from different experiments and put those together to project predictions, which we thought was pretty cool. So now I want to, and this is the first time I've talked about this stuff, but this is sort of taking a small part of this large collaborative project on Streptanthus and Colanthus, which is a collaboration with Jenny Greber, Julian Maloof, and Sharon Strauss on a Domestic Biodiversity grant, and focus on the seasonal germination in a changing climate. But just look how cool this is, with many flowers, maybe lots of cool morphology and so forth. And Sharon Strauss and Eva Lukacu have worked out a phylogeny for this group, which makes it easy to do phylogenetic analyses. Here I want to just ask, how will different species respond to changing rainfall timing? And so here again, we did an experimental manipulation of rainfall onset date, again, in the same screen house. And here's the team: Jenny Greber, Sharon Strauss, and our wonderful techs, Sarah and Arkell, and Sam Worthy, who's the postdoc whose beautiful analyses graphs I'm going to be showing you. And so again, we did 7 cohorts, planting cohorts here through the season. And you can see as we're planting them, seasonal temperatures are dropping. And cohorts 2 and 4 represent historic and current mean onset dates, respectively. And then we daily census these little seedlings for emergence, and did that until nobody came in the first year. And what we found is that the species do differ in sensitivity of germination timing to rainfall onset timing. 8 of the species germinate later with later rainfall onset. So as the temperatures get colder, they germinate less, but the slopes are different for some of these species. 3 of these species, actually, there's a difference between cohort 2 and cohort 4, which suggests that under present day conditions, they're germinating less on average than they would under historic conditions. And then some of these desert species really just don't germinate very much at all. And these are probably, just doing a little bit every year, dealing with what might be a bed hedging strategy. These species also differ in the fates of the seeds that don't germinate in the first year. So what we did, took all the pots where seeds didn't germinate, let them sit over the summer and bake, and then gave them a rainfall treatment early the next fall. And what you can see in some of them, for this species, glandulosus, that's for Tatiana, the ones that didn't germinate in the late season and the first year did come up a lot in the second year. On the other hand, some of them didn't have very much second year germination at all, which suggests maybe those seeds just died or went into very deep dormancy. And then here's one of the desert ones that germinates a little bit in the first year and germinates a little bit in the second year. It just seems to be hedging its bets by dispersing its seed germination across multiple years. So that's pretty neat. So what's ongoing now is looking at the diversification of traits like this on the phylogeny. So in this case, this is just looking at the slopes of these lines on a phylogeny to test for a signal of constraint, but also which is coming, but also to look for correlations with climate and with other traits on the phylogeny. So that's just, work in progress. A big question for this part of the project that hopefully we'll have the answer by this time next year is how does germination timing affect fitness in these species in a Mediterranean climate? In particular, are we going to see the same fitness declines with later rainfall onset in Streptanthus calanthus as we did in Arabidopsis thaliana? If this is a general pattern for Mediterranean annual species in California, then this could have pretty big implications for population demography. So we're analyzing the data from these experiments now. We do know for Streptanthus tortuosus that the timing of germination actually determines annual versus perennial life history. So if they germinate with early rains, this was a separate experiment, pretty much all of them will flower in the first year under our screenhouse conditions, but later cohorts will not flower in the first year. They have to then over summer, they have to parinate and can only flower the second year. And the early cohorts were much less likely to parinate, so they are effectively, most of them, annuals. The later cohorts then were biennials or potentially perennials, but they had to survive the summer in order to do that. So and we know also by experimentally manipulating that this is driven by a vernalization cue. So the ones that germinate too late don't accrue sufficient chilling hours to be able to flower in the first year. So we're looking forward to finding out results from more species, and we'll keep you posted. Just quickly, there's more work in progress by the team as a whole, just to give you the overview. We've been doing a lot of work with controlled chamber experiments, quantifying the hydrothermal parameters underlying germination niche variation. So the accumulation of hydrothermal time, temperatures, and also what water potentials that allow seeds to germinate. Julian Malouf and his student John Davis are identifying the genes differentially expressed by these different species in response to hydrothermal germination cues, and are, very excited to wait for those results. And then next step for Julian and Sharon is comparative analyses. So using the phylogeny that I showed you, testing for the association of gene expression phenotypes, germination niche phenotypes, and also climate of these species to test for whether there's constraints or how these correlate with climate. And then, both population genetic and phylogenetic tests for positive selection and evolutionary constraint for both genes and phenotypes. And then the kind of culminating project that's going to be grounded in part on the results that I just showed you will be demographic models, which would forecast population dynamics and vulnerability across species ranges under future climates. And that's led by Jenny, and Sam Worthy is the postdoc working on those models. So just summarizing then, I hope I've convinced you that the seasonal germination niche really is critical for climate adaptation. We can see a geographic signature of climate adaptation within species, both phenotypically and genetically, for traits in the germination niche. And we've also seen diversification of germination niche traits across populations of species. I've talked less about this today, but we do see natural variation in seed responses to environmental cues interacting with reproductive timing to shape life history, expression, and fitness. And so, the big questions remaining, which I hope you can see the motivation for answering, is more about what is the genetic basis of climate adaptation and diversification in the germination niche across species. How will selection act on germination niche traits across species ranges in the face of climate change? Can they evolve? Can they evolve fast enough to allow populations to persist in novel climates? And then that kind of leads into the question of what are the consequences of germination niche and its evolution for population fitness and ultimately for species distributions, especially in a changing climate. Okay, so many thanks. Just again, Alejandra really and Miki really spearheaded the screenhouse work that I showed. Ecological genomics collaborators, our dimensions collaborators, Jenny, Jewel, and Sharon, especially Sam's work I talked about today, and then the other postdocs, grad students, and techs, and many, many undergraduates that there wasn't room to acknowledge on this slide. And thanks to you for listening. And I'm happy to take questions.
Are there any questions? I guess you could either unmute or just type it into the chat if you need to. So Annie, amazing work, and I'm looking forward to seeing what's coming with the streptanthus stuff. I did have a question about the dog one stuff and Arabidopsis, and maybe I missed it, but why do you think it was that there's this signal of pleiotropy in previous studies, but not in the one that you did?
Honestly, I don't know. There was a weak signal in ours, so it may be just a power thing, but we certainly had power to detect lots of other things. So it's a good question that I don't know the answer to.
Okay.
Let's see. I might stop sharing my screen unless somebody wants to see it, because I see there's a chat that I don't seem to be able to. Yeah, there's two chats. Okay.
So it looks like Dan's asking, does later germination delay flowering in general, or does vegetative growth stop earlier?
That's actually a really good question. So depends on the timing. So in general, both in Arabidopsis and what we found in Streptococcus tortuosus is it does delay flowering, but not as much as not in a one-to-one basis. And the reason for that is later germinants, they're smaller, but as you get into the spring, days are getting longer, it's getting warmer, they grow faster and they kind of catch up. So they'll be later, but not as delayed as you would have expected based on how much later they germinated. Does that make sense?
Dan says yes, thank you.
Okay, I'm sorry if I went too fast. I guess that was a lot to assimilate.
I have a question. My camera's not coming on.
Yeah, go ahead.
that?
I've got a question about how close is Streptanthus to Arabidopsis? They're both in the Brassicaceae. And can you take advantage of any of the knowledge and Arabidopsis genome and that kind of things in those species?
Well, that is the goal. Yes, it's not super close, but it's all right. I'm trying to. It's not in the same branch, but it's close enough, and it does from what's Julian has been able to use that information to, we expect that some of these same candidate genes, they might be operating in different ways, but for example, we're really expecting that dog one would be worth looking at. So that was one of the reasons originally why years ago Sharon chose this group for her earlier investigations she started out looking at serpentine soil tolerance. And so, they're assembling a draft genome of Streptanthus diversifolius, Julian and collaborators. And so hopefully we'll be able to get at it in a much more genomic way in the future, or they will.
My second question is just out of curiosity, what's the pollinator of those species?
Super cool question. We see little bee. I would say if I had to pick one, like little native bees, but you certainly see honeybees as well. Occasionally see butterflies, but I think it's mostly bees.
Yeah, so then there's some interesting questions, right, about pollinator changes in response to climate and rain and all of that too, right? I'm sure that you guys are thinking about, or Sharon's thinking about maybe.
Well, and in tortuosus along an elevational gradient, already so one of Danielle de la Pasqua, who's a student of Jenny's, is actually looking at herbivory. Actually, also Elena Sulia, another student, has been looking at herbivory, but Danielle's looking along the elevational gradient and has hypotheses about how that's going to play out for plant traits, because there's dramatic variation in a bunch of traits along this elevational gradient, not just life history, but leaf traits, herbivore resistance, clearly just from our common gardens in the greenhouse, pest resistance, the high elevation populations are very mold susceptible. And then just dramatic phenotypic differences in terms of bushiness or leaf shapes or leaf color, flower color. All of these things are very diverse within species as well as across species. It's pretty cool.
Thanks.
Lots to work on.
Okay, Noel Mano has a question. He says, thank you for the great talk. Can you describe some of the species of animals or other plants that are heavily reliant on the current germination timing and would be negatively affected if germination timing was very severely altered?
Great, thanks, Noel. So I would say most California annuals are probably in this boat. And there's been a little bit of work. Jonathan Levine did some work in a field experiment showing that, I forget exactly the details, but I think it was a rainfall manipulation or a watering treatment, and showed, as we did, like reduced fitness for later germinating. I forget the species, but they were California native annuals. So I think it's probably a pretty general pattern, because if they germinate later and the growing season ends at the same time, then they just don't have as much time to accumulate resources. They might even run out of time for flowering and completing their life cycle. So for perennial plants, maybe it's less of an issue. I think they're less time constrained, and that they would continue through the summer. They have the adaptations to survive drought a little bit. But for annual plants, I think it's going to be a common phenomenon, resulting in lower population sizes. But that's just my speculation.
Okay, Josh Woodhelm has a question.
Yeah, actually, you just kind of started to say it right as I raised my hand. I was wondering too about translation to perennial systems. It's 72 degrees here today. I have a salvia bush in my front yard, and I was drinking a cup of coffee on my front porch earlier today, and I saw a bee flying around on the flowers, and I was thinking, that's a weird sight here in Indiana in November. And I was thinking about the, is this disrupting pollinator life cycles with, plants that are emitting volatiles at this time? And so I'm just curious, I mean, how much of this translates into variation within perennial systems? What are some of the caveats there?
Right. So perennials also, and it's also this, I'm focusing on Mediterranean plants, and I would say in Mediterranean climates, I think the perennials do germinate at the same time, but it's, that's kind of what happens in the first year. And so, I mean, we did do some work showing that it carried over a little bit to the second year in tortuosus, but it's more really, do they reproduce and survive in the first year, which does have huge impacts on population demography, but after that, once they're established, it's more just having to respond to all the other stresses of climate change, I think.
So at some point that memory's erased?
Yes, I think basically you can say that the early germination niche determines, like the offspring environment, determines whether it thrives or not. And that then translates maybe into future fitness, because it's not so big going into the second year or something like that, or might have a lower survival probability to the second year. But once it makes it to the second year, then I think that's less of a concern. Then there's all the other things you were just talking about, which is just, phenological disruption and flowering at the wrong time, and, emerging too early because the snow melts early and then you're hit by frost. And,
Yeah.
I mean, one of the things, that's something that's turning up in alpine systems a lot with snow melt.
Thank you.
Okay. I didn't have any other questions come in, so last chance to ask something.
Well,
I had one
Tatiana.
question. Hi. With like the rainy season in California becoming just like increasingly variable in between different years, in like your own opinion, do you think that these species of Streptanthus are kind of trying to hit like a moving target in terms of like adapting to different germination timing? Or do you think it's more of like they're adapting towards kind of like a median in between those like drastic fluctuations?
No, that's a really good question, and it's a sophisticated question. Because on the one hand, like, so how do I put this? So, I mean, on the one hand, like fluctuating climate that's been shown in trees and budburst might maintain adaptive genetic variation to allow them to respond to future selection pressure. So that's like an argument why this might help. On the other hand, it might, the fluctuating selection might not allow it to move towards the sort of long-term mean adaptive optimum, which might be bad. So I'm not answering your question. But I mean, this is one of the things hopefully Jenny Grimmer's demographic models will start to get at, is you can run scenarios where you have variation. What if you have a lot of dry years in a row, or a lot of late rain years in a row? Or what if they fluctuate? Sort of what will happen to populations under these different scenarios. So it's a good but complicated question. Thank you. So, okay, well, thank you very much. I've really enjoyed my visit, and thanks for the good questions. Cindy, am I staying on this line to talk to people, or do I go back to the?
We'll go back to the other one for your last meeting at 5.
All right, great. All right.
Thank you.
Okay, thanks. Bye-bye.